Hello, I am Dr. Suman. I'll be teaching you glycolysis today. So, glycolysis takes place in cytoplasm, and here we are using glucose to give us energy. What is the energy in the form of ATP, right? So, this path is common for both aerobic and anaerobic pathway. It is called EMP pathway or the name of the scientist Emden, Meyerhoff and Parnes. EMP. Hence, it is also called EMP pathway. Now, glycolysis means there is lysis of glucose to give us two molecules of pyruvate, NADPH2 and ATP. Also remember, NADPH2, one molecule of NADPH2 contains three molecules of ATP, right? Okay. Now, let us see what is the source of glucose. Source of glucose is photosynthesis. If it is sucrose, it will be converted by invertase enzyme into glucose. This glucose will be converted into two molecules of pyruvate, right? Now let us study the cycle. It's a very simple cycle. Just very coolly try to understand. First, we have taken glucose molecule. This gets converted to glucose 6-phosphate. Glucose is 6 carbon atom. Glucose 6-phosphate means one phosphate has been added. How is it done? Here there will be conversion of ATP into ADP, right? One ATP molecule has been used and the enzyme is hexokinase. Next, glucose 6-phosphate then comes to form glucose fructose 6-phosphate, right? Fructose, as you know, is an isomer of glucose, hence the enzyme is isomerase, right? After this, it forms fructose 1,6 biphosphate. That means one phosphate has been added. Again, how is it done? ATP molecule will lose one phosphate to form ADP. That phosphate will be added here to form fructose 1,6 biphosphate, right? After this, aldolase will come into action. It will cleave the compound. It will cleave fructose 1,6 biphosphate into two isomers, right? One is glyceraldehyde 3-phosphate. The other is dihydroxyacetone phosphate. If dihydroxyacetone phosphate is formed, it will again form glyceraldehyde 3-phosphate, right? These both are 3 carbon atoms, right? And till now, we have used 2 ATP molecules. That means minus 2 ATP. Let us keep this in mind. After this, we'll have the payoff. That means we will start gaining the ATPs. Now, ATP will be formed after this part of the cycle. Now, glyceraldehyde 3-phosphate will convert NAD into NADH, right? This is by the process of reduction, right? And glyceraldehyde, which is an aldehyde, that will be converted to acid by oxidation to form 1,3-biphosphoglycerate, right? This 1,3-biphosphoglycerate will form 3-phosphoglycerate, right? And here what will happen? Two molecules of ATP will be removed from 1,3-biphosphoglycerate. That means ADP will be converted to ATP. We will obtain these two molecules of ATP, right? So 3-phosphoglycerate will now be converted into 2-phosphoglycerate, right? By the enzyme phosphoglycerate mutase, right? Then enolase will convert 2-phosphoglycerate into phosphophenol pyruvate. Then phosphophenol pyruvate will get converted into pyruvate, which is again 3 carbon compound by the enzyme kinase, pyruvate kinase, right? And the cycle from here will take place 2 times because there is cleavage. One molecule of glyceraldehyde 3-phosphate, the same reaction will follow in the second chain. Hence, in this we see that 2 plus 2, 4 ATP molecules are produced and 2 molecules of NADH are produced, right? Now let us see the net gain and loss. First we come to net gain. Net gain means exactly how much ATP has been produced and how much has been used. Initially, till the payoff, we saw 2 ATP are used. So we have minus 2 ATP. After the payoff, we saw 2 ATP are produced. So that comes to be 4 ATP. 4 minus 2, the net gain shall be 2 ATP. Right? Now we come to the gross gain. In the gross gain, we saw that after the payoff, 2 NADH2 are produced, 1 NADH2 is equal to 3 ATP. So 2 will become 6 ATP, right? And there are 4 ATP also produced. Hence our total will be 4 plus 6, that will become 10 ATP. But initially we saw you have to take this also into consideration. For the gross, 2 ATP are used. So it will be 10 minus 2, 8 ATP is the gross gain. Gross gain is 8 ATP and the net gain, that is only ATP. We count only the ATP in the net gain, that is going to be 2 ATP. Right? After this, what happens to the pyruvate? The pyruvate can either enter Krebs cycle if it is having oxygen. If there is no oxygen, then it will undergo fermentation, form lactic acid or ethanol. Right? So this comes to the end of glycolysis. Thank you.